Good evening, everyone from India. I'm Professor Dr. Seema Bhatnagar, Assistant Director at Amity Institute of Biotechnology. And today I'm here to talk about folate receptor, a rational therapeutic target for cancer treatment. This is the area in which I'm working. And I will also be speaking a bit about the programs at Amity as per the session schedule. So cancer, as we know, is becoming a global epidemic, and we are seeing more and more cancer cases. It is estimated that 1 in 5 people will be suffering from cancer in coming years, and this includes 1 in 8 men and 1 in 8 females will also succumb to the disease. So this is something really alarming and something that requires immediate attention. And so that is what is the focus of my talk today. We are also seeing that in middle and medium and low income countries, there will be a steep rise in the cancer cases as predicted by 2040. As compared to the countries where already there are more cases, but in future we will see more cases in countries which are of low and medium income. So India falls in that category, and that's why this is a matter of concern to everyone. If we talk of the cancer mortality, the situation is alarming as well. So if you see the gender divide, males and females equally are equally prone to the disease, and we are seeing a high mortality of the disease both in males and females. And India has a high mortality rate. So historically, if we look at the drugs or the chemotherapeutic agents which were available for treatment of cancer, we see that the first attempts to look at chemotherapeutic agents started somewhere in the late 1940s, and truly the decade between 1960 to 1970 can be called as the decade of adjuvant chemotherapy, where a number of agents were not only discovered, but they were also tested, and many of them were from plant sources, and they are still in use today. However, they have a number of drawbacks, and that is the reason for the growing concern about how we can improve the quality of life of individuals and how we can prevent the disease, or if not preventable, at least treat them and improve the quality of life of cancer patients. The last 20 years from the year 2000 has seen a large number of small molecules that are coming up for targeted cancer therapy. There are a number of targets that have been explored, and small molecules that target these receptors or pathways for cancer have shown a lot of promise. However, if we look in terms of the experience of patients, we don't see much improvement. And according to the World Health Organization, every year in India, at least 10 lakh individuals suffer from the disease. And the spending in a cancer suffering household is at least 36 to 44% higher than as compared to households of similar demographics. Even the cost of treatment has gone up. So what is really wrong? Are we not having drugs? Or if we are having drugs, then why are we not able to reduce the cost? And what is the way to improve the quality of life of individuals who suffer from the disease? So this is something that needs to be looked into. So all this has given initially, there was the focus was on traditional medicine, which included radiation, chemotherapy, and surgery. But now this has given way to more targeted therapies, the involvement of genetics of individuals, and also immunotherapy for treatment of the disease. So our group at Amity is involved in study of the folate receptor, and we are looking into new drug delivery options, how we can deliver low concentration of the drug to the cancer cells in a targeted way. And the folate receptor appears as a very attractive target for cancer drug delivery. The reason being that, first of all, it has been newly identified, and there are 3 isoforms of the folate receptor alpha, beta, and gamma. However, the folate receptor alpha has seen a lot of success and a promising therapeutic target in a variety of cancers. Non-malignant tissues express very limited amount of folate receptor alpha, whereas the malignant tissues, and especially solid tumors, the ovarian cancer, kidney, lung, brain, breast cancer, lung cancer, and you name it, all these have overexpression of the folate receptor. Also, what is important is that the folate receptor alpha has high affinity for folic acid, and conjugates of folic acid can be considered as good opportunities to target the cancer cells. And a number of clinical trials are also ongoing in this area. So I will spend some time talking about the accessibility of the cellular uptake through the folate receptor. We know that 80% of the human cancers are derived from epithelial cells, and those receptors that are restricted to the apical regions of the cells can be considered as good targets, because when drugs are administered parenterally, they are not accessible to normal cells. And this is a big advantage, because most of the drugs that were already available, they lack selectivity, especially the chemotherapeutic drugs. They lack selectivity. They show a lot of toxicity, because they not only target the cancer cells, they also target the normal cells. So if you are using a targeted approach, then obviously you will be able to take care of the off-target toxicity, which is seen in most cases. So since receptors are apically expressed, so when the drug is administered parenterally, it is not accessible to the normal cells. However, when the cells become malignant and the tight junctions collapse, in those cases, the folate receptor becomes redistributed on the cell membrane, and that is the reason why it becomes accessible to drug conjugates which may be administered parenterally. So this is a big advantage, that where you can opt for targeted drug delivery. So 3 general strategies have been explored. We have anti-folate receptor antibody approach, the humanized folate receptor alpha antibody drug conjugate approach, and the folic acid SMDC approach. SMDC is the small molecule drug conjugate approach. So if you look at the molecule of folic acid, it comprises of 3 regions. You have the pteridine ring, you have the para-aminobenzoic acid portion of the molecule, and you have the glutamic acid. So it is the pteridine ring which is primarily involved in the binding to the folate receptor. Therefore, this part of the molecule is very much available for functionalization and can be used for drug delivery. So a couple of drugs just as examples. So here we have the antibody approach, where farletuzumab has been tested not only in phase 2 but also in phase 3, and what has been seen so far is that it did not meet the primary endpoint of improving progression-free survival in patients. There are also some studies which show the antibody drug conjugate approach, and MIRV is one such molecule, one such agent which has been tested, and it is showing some promise, but still in phase 1 and phase 2. Then we have the folic acid small molecule drug conjugate approach. So the folic acid conjugates can be used to deliver the drug payload to the cancer cells. So, what it involves is that folic acid can be conjugated using um, cleavable bonds, and these can carry the drug payload to the cancer cell. Once the folic acid binds to the receptor, it is internalized, and the internal pH of the endosome will result in the cleavage of the folic acid from its receptor and also release the drug payload. Now, once the drug payload is released, the folate receptor recycles back to the cell membrane, and the drug is delivered at low concentration to the cancer cells. So that is a very interesting approach, and our work is in that direction. However, we have made some changes which I will be talking about. So vincafolide is one such molecule where they have tested a vinca alkaloid and they have tried to deliver it to cancer cells. This molecule showed a lot of promise. However, it is now out of phase 3 trials. So keeping all this in mind, we looked into the literature, and curcumin, which is a well-known spice which is used in Indian food, and it has a lot of medicinal property. We looked into this molecule. It's a wonder molecule because it has a whole range of activity and shows a lot of promise, but there are some things which have to be taken care of. So it has been associated with antioxidant, anti-inflammatory, anti-cancer, antiviral, antibacterial properties. It is also known to chemosensitize many chemotherapeutic agents such as 5-fluorouracil, doxorubicin, paclitaxel, cisplatin, etc. The only drawback of this molecule is its poor bioavailability and poor absorption. So that is what limits its use in the clinical setting. So keeping in mind the multiple targets that this molecule is known to modulate, we have designed our own small molecule drug conjugate, which comprises of folic acid. There is a spacer unit which connects it to curcumin, and finally there is a cleavable bond that is connecting to an anti-cancer drug, and we have used paclitaxel. We have used acid-sensitive cleavable bonds, which can cleave in the acidic pH of the endosome. So I'm going to share some of the results, preliminary results that we've got with this triple conjugate that we have designed. We have tested our conjugate in the cervical cancer cell lines like Hela and SiHa cell lines. And what we find when we do the MTT assay of these compounds is that our conjugate is working as good as the known drug doxorubicin, which is really important because curcumin might be assisting in its in the drug entering the cancer cell. The cell proliferation assay also, when we compare it with curcumin, doxorubicin, and with untreated cells, we see that it is showing comparable activity at 12 and 24 hours, both in Hela and SiHa cell lines. In order to look at its ability to enter the cells, because it's a large molecule, is it able to really enter the cells? So we have carried out these studies where we have seen that our molecule is able to enter the cells, and this green fluorescence that you are seeing is of curcumin, which has entered the cancer cells after treatment. We have seen it at 6 hours and 18 hours using not only curcumin but also the conjugate. And our conjugate exhibited better fluorescence as compared to curcumin, which is evident from the figures. We also carried out some in vitro cell migration assay using our conjugate. doxorubicin and curcumin and with the controls. And what we found is that in case of the untreated cells, whereas there was 100% migration of cells, but when we treated with either of the agents, we could see that cell migration was very much inhibited by using the conjugate specifically. So we've also compared the apoptosis of we have carried out the apoptosis assay with our conjugate curcumin and doxorubicin in the same cell lines. And we find that this molecule, this conjugate, in fact, shows the cells are more apoptotic when we treat them with the triple conjugate. So just to summarize what we have got so far is that a conjugate is exhibiting better cytotoxic activity as compared to curcumin or doxorubicin alone. There is 30 to 70% reduction in cell proliferation in both Hela and SiHa cell lines. In our uptake experiment, we did see that the conjugate could internalize in the cell, and we have seen it visually that more concentration of the conjugate or curcumin, which is could be seen inside the cell, which means that indeed the conjugate was internalized inside the cell. The scratch assay again showed significant reduction in the migration rate as compared to the untreated controls, and the apoptosis assay after treatment with curcumin doxorubicin showed that the apoptosis rate was much higher when we used our conjugate. So all these studies gave us a lot of confidence, and now we are proceeding ahead with more in vivo studies for our conjugate, and we believe that curcumin would be the game changer for this conjugate, because it is assisting in the entry of the drug by chemosensitizing the cell. Of course, more studies are required for it, but our preliminary results indicate that probably curcumin is assisting and the drug is able to enter the cell at low concentration. So if it is able to enter at low concentration, we are able to also target the cell. So we expect that the off-target toxicity will be taken care of, and at low concentration, we are expecting similar results as compared to the known drug. So my next part of my presentation is about the graduate and the postgraduate programs at my department. So we are Amity Institute of Biotechnology at Amity University, and we offer a lot of programs in the area of biotechnology, which includes BTech, BTech Bioinformatics, BSc programs in biotechnology, BSc MSc Dual, BTech Dual 3-continent programs, and we also offer BSc Biotechnology and B.Ed courses. And for our postgraduate programs also, we are offering the MBA Biotechnology program, MSc Biotechnology, Bioinformatics, MTech Biotechnology, and of course our PhD programs. We also have some certificate programs in artificial intelligence. We have Masters of Technology in Bioinformatics. So it's a big focus in our department, and we have a large number of students who are really interested in all aspects of biotechnology. Our institution also offers many opportunities for international opportunities for students, which include the student exchange programs, which are offered in France, where students can spend a semester abroad and complete their studies in a foreign university. We offer study abroad programs at 4 or 5 locations all over the world, and these are available to all students, UG and PG students at Amity Institute of Biotechnology. We also have a very unique 3-continent program. Now this program, we have 3 streams in which we run them: the BTech Biotech 3-continent, BSc Medical Biotech 3-continent, and the BSc Honors Biotech 3-continent. So in this, the students complete most of their studies in India, but in the last semester they travel to UK and US for 3 months each, and finally they get their degree from Amity University. We are also collaborating with many universities abroad, and we are offering international collaborative programs in BTech Bioinformatics, BTech Biotechnology International. Again, it is a 2+2 program with Indiana University, and the BSc Honors Biotechnology program is with the University of Queensland, Australia. It's a 1+2 program. So students have a plethora of opportunities if they join the department, and truly we believe in creating global citizens at Amity, and our students have always made us proud. So and we hope to continue the same in future. So thank you very much. Thank you for your patient hearing. If there are any questions, I am ready to answer them.
Thank you very much for this presentation. We do have a few brief questions here. One of them are there any specific techniques required to synthesize these kinds of molecules? He means conjugate.
Yes, so conjugation chemistry would all involve chemical synthesis of the conjugates, but yes, it's very important to characterize whether conjugation really took place or not. So of course you would be using IR, NMR, and you have to use, since it's a large molecule, so you may have to go for MALDI-TOF to know the molecular weight of the molecule. So all that is very important, because just carrying out the synthesis does not mean that the conjugate got made. You have to characterize it.